Okay, I'll go ahead and get started with an introduction, and that'll take a minute or two as we have more folks joining us. So welcome everyone to the UW Data Science Seminar. As you all know, this is a seminar where we showcase data science in the arts, engineering, humanities, and sciences. We're delighted today to have Steve Peterson joining us to give a talk. And Steve is a postdoctoral fellow who's co-advised by Bing Brunton and Raj Rao. And he's currently studying electrocortical, oh goodness, Steve, you say it.
The mouthful. Yeah, electrocorticography.
I knew I was gonna screw it up. During naturalistic movements. And he received his PhD in biomedical engineering from the University of Michigan, where he performed high density, I'm gonna get this one, electroencephalography, pretty close anyway, recordings during perturbed balance beam walking in the lab of Daniel Ferris. And his interests extend to real-world neuroimaging, neurosignal processing, and cortical integration of sensory information. He's one of our UW Data Science postdoctoral fellows as well, which is a title that the eScience Institute gives to data science postdoctoral fellows, to current University of Washington postdoctoral fellows whose work extends into using data science tools and techniques across a broad range of application spaces. So we're delighted to have Steve talking with us today, and I will turn it over to you. I guess just a quick note, if you have questions, please drop them in the Q&A, and we will address those at the end of the talk. But you can put questions in at any point, yep, I'll turn it over to you.
Thanks, Sarah. Yeah, so as Sarah mentioned, I'm a postdoc in Bing Brunton's lab in UW Biology, and I'm co-advised by Raj Rao in CSE. And so I'll be talking about some of my research doing naturalistic neuroscience and neuroengineering. And just a brief little background. So I did my undergrad at Washington St. Louis in biomedical engineering, also did my PhD in biomedical engineering. So my interests and my training have really been kind of at that intersection between life sciences, specifically neuroscience, and engineering, and using data science tools to understand and ask interesting questions about the brain. And as an undergrad, what really sparked my interest in research was actually being able to do research on insect olfaction, so very different from what I do now. So for that, I actually got to work on some projects doing odor conditioning, similar to the classic experiment where you ring a bell and have a dog salivate to the smell of it, to the sound of a bell, because they expect a reward. But in this case, it was an odor. And also getting to create this T-maze experiment to test odor preference in locus, and then see how that translated to neural activity responses. So in this talk, what I transitioned to in my PhD and my postdoc has been looking at humans during different types of movements. And in general, people make many different types of movements. We may not be making that many right now due to the pandemic and since we're all at home, but in general, we make a lot of really complex coordinated movements. However, so that's what happens in the wild, so to speak. But when we get to studying movements in the lab, we often study very restricted movements because, for example, we have MRI machines which give you really good resolution spatially in terms of recording neural activity, but require you to lie very still. Additionally, a lot of experiments that you get in the lab tend to be very repetitive. So they tell you to kind of put your hand in the center, move to this corner of the box, and just keep doing that, or move to this other corner. And in reality, our movements are much more unstructured and spontaneous. So the goal of my research in general, in both my PhD and my postdoc, has been kind of to move from this side on the right to more towards what happens in the wild, and being able to understand what goes on in the brain and understand the neural basis of more naturalistic movements. But, I just told you that you can't really use, something like a fMRI scanner. So how do we actually measure neural activity during such naturalistic movements? And the way that I've done that in my research has been to look at neural population recordings. And so these involve intracranial electrocorticography, or ECOG, which are electrodes on the surface of the brain underneath the skull. Additionally, I've also looked at high-density scalp electroencephalography, or EEG, and these are electrodes above the skin, so not underneath the skull. And so for ECOG, this tends to be a very invasive recording, so you have to remove a piece of the skull to actually put the electrodes in. But because the electrodes are on the surface of the brain, you get a really nice clean signal that you can record from, at least compared to EEG. However, it's also clinically motivated, so you're only performing this procedure on clinical patients who need it for stuff such as epilepsy monitoring because it requires surgery. Alternatively, EEG is much more non-invasive, so you don't have to remove a piece of the skull to record data in that case. It has very wide spatial coverage across the entire top of the head, but the signal is a bit more noisy compared to ECOG. And this is because you have muscle activity in your neck, eye movements, and even just motions of the head that can really contaminate your signal potentially, depending on the type of task that you're doing. But also just comparing both of these to something like fMRI, which has really great spatial resolution and really good reliability, both of these methods suffer from some level of lower spatial resolution and also noisier signals that can be potentially less reliable from one trial to the next. So this is sort of a limitation of using these analyses of these recording modalities when we want to use these, especially for naturalistic movements where the data can be much more noisy than traditional experiments. So the idea, one of the big things that I've leveraged in my research is kind of using these neural population recordings while also applying data science tools to really address some of these shortcomings in terms of the spatial resolution and noise of the signals. And by doing that, it can really get a handle on and really get a look at naturalistic neuroscience and naturalistic movements and understand the neural basis of them. So specifically, I'm going to talk about, I'm going to emphasize really two data science tools that I've used a lot for my research. One is independent component analysis, or ICA, and the other is using neural networks. And so independent component analysis is the idea that the idea with independent component analysis is that you have these neural generators in the brain that are actually generating the activity that you record at the electrodes. So those are shown by the blue dots here. And you're recording activity at the electrodes. It's actually a linear mixture, or you're assuming it's a linear mixture of these blue dots. And while that may or may be somewhat of an oversimplification, it actually works really well in an assumption in this case. And so what you do is you take this data that you're recording that's a mixture of these sources that are neural and potentially non-neural as well. And what you do is you can apply independent component analysis to basically return these recovered signals that are independent, statistically independent over time compared to each other. And the hope is that these signals actually recover some of these original generators within the brain well. And so when you actually do this and apply it to high density neural electroencephalography data, what you get for each independent component is you get a trace, which is basically a linear mixture of the electrode data that you had originally for each component. And then you can also get a spatial location that's the approximate best location of that neural trace, of that independent component. And basically when you do this for some data, you can actually show that you get real physiological signals that you'd expect and can measure by putting electrodes near the eyes or near the neck. And so in this case, this is showing that you get eye blinks. So these trajectories really correspond well to eye blinks. So you can also get saccades as well. And both of these show up near the eye as you would expect. You can also get heart rate, or EKG. In this case, the model doesn't really know where the EKG would even be, so it just kind of throws it off to the side. But additionally, you can also get left and right neck muscle activity. And this is really important because it's a major contaminant of EEG data in general. So a real world example of using ICA during movement is shown here. And this is a fairly older paper by my PhD lab, where they recorded high density neural EEG data during walking on a treadmill. And what they found, and these are the independent components across multiple participants, but basically what they found are these green, red, blue, and magenta dots indicate neural independent components that they found. And what you can see too is for each independent component, the big dots indicate the centroid or the average position across all of these little dots. But you can see for each little dot, you actually get really nice spatial location in terms of, you get a better idea of where that activity is coming from, and much better than you get from just saying, oh, I'm recording activity up here, so it must be right there, because you can get mixtures from sources all over the place. So this improves your spatial localization, but it also improves or reduces the amount of noise in your signal as well. Because in addition to getting these neural sources, you also get all these yellow dots. And in this case, the yellow dots indicate independent components that are actually muscle artifacts from the neck. So IC is really helpful for denoising your signal and improving spatial resolution, particularly in EEG. The other data science tool I'll talk about is our neural networks. And the idea here, as many of you know, you have a bunch of nodes and weights. And the idea is that you can train your weights using data-driven, based on the input data you gave it and labels that you expect it to show at the end. And that model will actually learn how to appropriately find that label given the inputs that you have. And so this has actually been used quite a bit for electrocardiography data, as well as EEG data. And one of the benefits you get with this is you can actually do really complex neural decoding with neural networks. And this is because they pick up these really complex spatial patterns in the data, as you can kind of see here. I mean, to me, this looks like just gibberish in a lot of ways. But you can, with this complex pattern, you can actually robustly decode speech. And this is really useful too, because each time someone talks and says a word, things can be, the neural signal can be a little bit different. The way they talk can be a little bit different. So neural networks seem to have this ability to kind of find robust features, depending on how you train them and all that. So those are kind of the two tools. So using these neural population recordings, in addition with these data science tools like ICA and neural networks, you can really get a handle on what's going on with naturalistic movements. And so for this, I'll talk about two specific projects that I've been working on that actually address each of these limitations in how we study behavior in humans. And so the first one addresses this issue with being able to record mobile movements and looks at the neural response to balance perturbations. And the other experiment I'll talk about deals more with looking at movements when they're undergoing structured trials and repeated trials, and looks at developing flexible generalized decoding during unstructured arm movements. So first, I'll talk about the neural response to perturbed balance. And so this is work I did as a PhD student in the University of Michigan. And so the real question we asked ourselves for this project was, when do you actually know that you've lost your balance? And I'm sure this happened to all of us where at some point we slip and fall, even happens to Shaq. But as you age, slipping and falling can have very serious consequences, and can really lead to loss of independence. So really understanding when people realize that their balance is lost could really help with patient diagnosis and even treatment, in addition to just being a really interesting basic science question. So in terms of human balance control, you can think of it as, you have sensory input from your visual, vestibular, proprioceptive, mechanoreceptive systems. And all of this feeds information into your central nervous system, so your brain and your spinal cord. And from that, it generates a neural command to your muscles to execute a certain type of movement. And from that, you change your posture, hopefully for the better, so you don't fall over. And most importantly of all, this is all a loop, right? So this happens in real time constantly. And it has to be very precise, has to be coordinated, has to be accurate. And so I was interested in sort of probing this interaction between the sensory information and the sensory integration processing in the central nervous system, because I can record what goes on, at least at the cortex level in the brain during sensory integration. And I can also basically perturb sensory inputs to on this side of the equation. So for this, I recorded EEG during balance beam walking. And this is a close-up view of what high-density EEG cap looks like. So each one of these little dots here is a different electrode. And so the cap I used is a little bit different. It doesn't have all these electrodes down by the neck. They're all up at the top of the head. But it's very similar. And I used 128 electrodes for each participant. And they were walking on a treadmill-mounted balance beam. So it was basically an infinite balance beam. They can never stop. But at the same time, they were always in the same spot. So that way, if they fell over very far, this would catch them before they got hurt. In addition, the actual recording device for the EEG system was up here. So the cables were out of the way. And so for this, I looked at two types of sensory motor perturbations. So one was looking at a side-to-side pull at the waist. So basically you have these bars on either side and one will pull at random times the participant a little bit to the left or to the right to make them have to adjust. And then I also looked at visual rotations. And so here's an example of the side-to-side pull perturbations. So you can see that there's this little bar right here, and every once in a while it'll activate and pull the person. You may not see much of an adjustment here by the participant, but there is still quite a tug that you can feel. Additionally, for the visual rotations, this was using a virtual reality headset and a little webcam underneath. And basically what that allowed us to do was have them view what the webcam was seeing, and then we could manipulate their visual field of view. And so what we did for this was we rotated their field of view 20 degrees clockwise or counterclockwise. Counterclockwise or clockwise, and had them have to adjust while walking 1 foot in front of the other on this little beam. And you can kind of see what that looks like here. And additionally, this is, while I say it's a rotation of your visual field, it's also instantaneous. So it just, it's at 0, and then the next split second, it's like this. And then it just goes back after a half second. So these are two ways to sort of look at the balance response in people as they're balancing, the perturbation response as they're balancing. And for this experiment, we actually looked at those two perturbations, but also while they're standing and walking. So there are 4 conditions, 10 minutes each, and we tested 30 healthy young adults that were gender balanced. And from those 30 participants, we found 8 clusters of independent component sources. And so these are sources where more than half of those 30 participants had an independent component that was in that location or nearby when we clustered it. And so what we found from that analysis, the ones we found were left occipital, right occipital, posterior parietal. So a bunch of ones towards the back of the head, which would be expected given that there was a visual perturbation. So you have to have a visual response during this process. Additionally, we found ones more in the central motor areas, as well as the anterior cingulate. And so all of these, are pretty consistent across participants, but also make a lot of sense given the task that we're performing in this case. And so with these clusters, we actually looked at the time frequency response to each type of perturbation. And so this is the response to the visual perturbations during walking. And so to orient you to these types of plots, the x-axis is time, where 0 indicates the onset of the perturbation. So in this case, this is the onset of that visual rotation. And then 0.5 seconds here, this other dashed line indicates the offset of that perturbation. So where it rotates back to 0 degrees. And the y-axis is frequency in hertz. And then the color mapping indicates power in each time frequency bin relative to the time period before the perturbation happened. So between -0.5 and 0. And what might strike you right away is that there's a lot of activity in these three areas right here. So left occipital, right occipital, posterior parietal, and all three of these are in the back of the head. And so, as you might expect, when you rotate and disorient the visual field a little, or your visual information a little bit, your visual areas towards the back of the head tend to be more affected. And so, yeah, and also we see this pattern where there's this low frequency power increase, so between about 4 and 13 hertz, and this higher frequency power decrease, so between about 8 and maybe up to about 30 hertz. And so when we look during the pull perturbations, when they're being tugged at their waist, we see a slightly different response. So a lot of the right, left, and posterior parietal areas are not as active as before. In contrast, though, all of these central motor areas, so all of these areas over here that are involved with, that are consistently thought to be involved with motor planning, motor execution, are a lot more involved, as you might expect. And especially the supplementary motor area, where there's this really large, low frequency power increase. Additionally, when you look at the right and left sensory motor areas, you see that there's this low frequency power increase, but also this, it's a little bit weaker for sure, but you still see this higher frequency power decrease. And when you do this during standing, when you look at the response to the pull perturbation during standing, it's much stronger, or I guess much more negative in terms of the values in this higher frequency power range. And so what's interesting about this response is actually very similar in some ways to this other response that you see during the walking condition, during the rotation condition. And so this led us to think that maybe there's this consistent perturbation response in the brain that differs based on which sensory motor areas are involved and which sensory information is perturbed. And to sort of explain that low frequency power increase and higher frequency power decrease in the context of literature a little better, I'll explain each of those parts separately. So the low frequency power increase corresponds really nicely to the study done where they had participants stand a little bit tilted forward while they were hooked up to this chain. And once they released the chain, they had to try to balance without stepping their foot forward. And so when they did that, at time 0, you see this really large, this really prominent low frequency power increase relative to before that happened. And this is in channel FCZ. And this is just a diagram that shows about where channel FCZ is relative to the supplementary motor area, which is where we saw our strongest activation. And this lines up pretty well. It's not perfect since it's a channel level activity and not source level activity using independent component analysis. But overall, this lines up really well with the literature and indicating that when you have to make a balanced response, an active balanced response, that this area seems to be very active. Additionally, this higher frequency power decrease corresponds really well to literature showing that when you kind of change states from sort of being in this idle state to having to actually do something or have to think about certain things, that you see this alpha beta decrease in power. And so this is an example of a study where basically participants were walking on a treadmill to the beat of a metronome. And at time 0 here is when they started delaying the interval between metronome beats. And so as the participants became aware of that, they had to adjust their gait to match it. And then they also exhibited this really long-lasting low frequency power or higher frequency power decrease. And it's also notable that this is in the parietal area as well. So the results we see make a lot of sense based on literature and based on how you respond to balance and how you respond to new information as you get it. What's also really important to note is that the neural responses don't really match the neck muscle activity that we see, which is great. So during the rotations, there's really no time-locked muscle activity at all. So these are recorded from the actual neck muscles themselves. But when you have the pull conditions, there is a lot of muscle activity that shows up and can contaminate our signal. But what's really nice to see is that during the pull perturbations, the occipital areas weren't as active anyway, and those would have been the areas most influenced by the left and right neck muscle activity. So it's nice to see that we're getting a clean, fairly clean neural signal given the mobility and the amount of movement participants were doing during this type of experiment. Another interesting comparison in the literature is that a similar pattern has actually been seen in the subthalamic nucleus as to what I was showing here. So this is an example, this was found during an experiment where they were looking at visual incongruency during a task where you have these 5 arrow-like symbols, and every once in a while that middle arrow will be pointing the opposite way of the others. And when that happens, you have this sensory motor conflict, and this sort of activity was found to be in this subthalamic nucleus. And you kind of see that this actually matches this low frequency power increase and this higher frequency power decrease quite well. So, I mean, there's no clear connection with this, but I think it would be very interesting to sort of probe this response that we see in EEG in people who have more balance impairments or, stuff such as Parkinson's, where you have these motor impairments alongside of other pathologies, because using EEG is much less invasive than actually probing into someone's subthalamic nucleus. So that seems like a potentially interesting area of research. Additionally, we did a follow-up study on this data looking at the information flow from one area to the next. And this was using Granger prediction analysis. And so the idea here is that, so these are all 8 of those clusters of independent components across those 30 participants. And so blue indicates a significant decrease in information flow between 2 regions, and red indicates a significant increase in information flow between 2 regions. And this is just right after the rotation occurs, the visual rotation. And this is in the 8 to 13 hertz band, or alpha band. And basically what struck me about this was that while while some of these are not as strong as others, it's very consistent that the 3 areas in the back of the head, so the left occipital, posterior parietal, and right occipital, each have these significant decreases in information flow between their corresponding parietal area clusters. And this is interesting because it kind of aligns well with the sensory reweighting hypothesis, where if the information that you get from your sensory system is not useful to what you're trying to do right at that moment, your brain might say, well, I don't want that right now. So it will downweight that activity. In this case, when you have visual rotations and you're trying to balance, having a rotation is not super helpful for trying to balance. And so this is interesting. So from this, we were actually interested too to see, can we actually use perturbations to improve balance training? And so this is some work that a grad student in my PhD lab, so Dan Ferris's lab, worked on quite a bit. I had done some initial stuff using virtual reality. And in this case, she actually uses occlusion glasses, which basically cover your eyes at predetermined times. And so the idea here is to actually train mostly your proprioceptive inputs and try to not train so much your visual, basically have you not rely so much on your visual input as you're trying to learn to balance on this beam. And what she found from that, at least so far, is that visual occlusion training actually improves your performance really, really drastically when you're doing the task, when you're learning it the day of. But also if you bring someone in 2 weeks later, performance is still not bad. It's not significantly different from unperturbed vision, but you can see that there's definitely some increase at this point. So it seems like potentially a promising direction in terms of using perturbations to train balance in addition to studying how the brain works. So now we'll go back, so kind of go back to that question I asked at the beginning, which is when do you know that you've lost your balance? And I think, this this perturbation response that we see within the neural data gives us an interesting way to sort of probe at this question without actually, making people lose their balance, and doing it in a non-invasive neuroimaging way. And additionally, it seems like it could be potentially useful for balance training as well. So now I'll move on to the second project I'll talk about, which is developing generalized decoders for unstructured arm movements. And so this is work I've done as a postdoc at the University of Washington. And so the idea here is to develop neural decoders for brain-computer interfaces. And the idea with the brain-computer interface, you have activity that you're recording from a patient's data, or from a patient's head, such as this, and you take that data and you model it, decode it, and generate a useful signal to control some type of device. So in this case, they're controlling a robotic arm. You can also use brain control to type on a computer screen fairly quickly. And these are really cool, demonstrations of what's possible with neural control, but there are definitely some limitations. And one of the big limitations here is the amount of training data you need to create a model and potentially retrain that model over time as the signals change and potentially as the behavior changes as well. And this isn't necessarily very practical when you're looking at real world unstructured activities where things can change all the time and the neural signal in and of itself can change quite a bit too, potentially. And so the problem here is that the training data that we can get for these decoders is often very limited and often very time consuming to obtain. You can't just do Google image search and take a bunch of images and train your decoder, at least not in this case. So one solution to this is to train on pooled data. So take data from multiple participants, and even if you don't have a lot for each participant, together and combined, you have a lot more than you normally have. And then when you train this generalized decoder, it should be more robust and have a nice, good representation that's not just tailored to the training data set that you give it. And you can then fine tune it using just a little bit of new data that you get. However, to do this, it really requires you to have a decoder that's robust across participant differences. And so what actually differs from one person to the next? And so one thing that might differ is the frequency content of the signal that you're recording. And so this is an example from our naturalistic movement data that we have when participants are just spontaneously moving their contralateral limb, their limb contralateral to their ECOG recording hemisphere. And so time is on the x-axis here with 0 indicating when they start to move their arm. And then frequency is on the y-axis. And then the color mapping, like before, indicates power at each time frequency bin in term relative to before they move their arm. And what you can see here, so this is average across 12 participants. You can see that there's a very consistent spectral pattern between, there's this higher frequency power increase. So it's flipped this time, a lower frequency power decrease. And you can see this is pretty consistent, or it looks very consistent. But when you actually look from one participant to the next, it's actually quite a bit of variability. There are some participants where this low frequency power decrease doesn't really appear at all, and several where this high frequency power increase is just shifted over in other ways. So it's really important when you want to build a decoder that can robustly decode across multiple participants to have it be able to account for these differences across participants. Additionally, another thing that can differ from one person to the next is the electrode placement. And this is particularly problematic for ECOG because it's based on clinical need, and the electrode locations vary from one participant to the next, and they're not standardized, as they should be because it's based on clinical need. So this is an example of electrode placement for 4 different participants, where each participant's electrodes are shaded a different color. And you can see that there's not always a great way to align disparate electrodes from each participant. So the main goal of this part of this project is to take data from multiple participants, train a generalized decoder, and then fine tune that decoder on a small bit of data from a new participant. And so for the generalized decoding part, we started with a convolutional neural network. And so we actually used a pre-existing convolutional neural network that works for EEG data, and it's called EEGNet. And so the idea with EEGNet is you have your EEG data on the left here, and you put it into this model, and it'll spit out a label. And hopefully that label corresponds to the behavior that was going on during that EEG activity that you gave it. And so there's definitely, I'm really oversimplifying what a convolutional neural network does here. But I'm focusing on two really important parts of this decoder because the convolutions that EEGNet does and any neural network can do are really interesting to me because they actually mirror real neural signal processing techniques that we do in every, when working with EEG and ECOG anyway. So one example is doing a convolution over time. And in this case, that would be separating the neural data you have by different frequency bands. And then from that, you can do, it does spatial filtering. So it basically finds which electrodes are most helpful for determining the correct output label. And it does that for each of these different instances of frequencies. And then it does a lot more processing after that I won't get into. However, it doesn't necessarily address these interparticipant differences as well as we would like. Specifically for the frequency content, it does have different, filter out the data into different instances of different frequency bands, but it doesn't actually convert it into the frequency domain where things are much more separable or tend to be more separable with neural data. So what we did is we added a explicit conversion to the frequency domain. And so by doing this, you have your time domain signal here, but then everything after this is in the frequency domain where things are much easier to decode between. Additionally, though, it doesn't account for this difference in electrode placement because it's made for EEG data, not for ECOG data. So if you give it an array of electrodes, it's assuming electrode 1 is the same for every single participant. And this works really well for EEG because electrodes can be very standardized. But for ECOG, that is just not the case. So what we did in that case was project onto common brain regions. And so the idea here, to use a simple toy example, is that if you want to estimate let's say you want to estimate what's going on in this brain region right here. And so you have these 4 electrodes nearby with which to estimate the activity. And so one reasonable way to estimate the activity would be to say, I'll take a weighted average of these 4 electrodes, but this red electrode here is about twice as close to these other 3, so I'll weight it twice as much. And so basically, you can do some sort of weighting like this. In reality, what I do is I drop a Gaussian on it, so it's actually, the red one would be a lot more weighted because it's closer. But it's generally the same sort of principle where it's based on distance. And obviously this sort of, the limitation of this approach is that it doesn't account for the actual anatomy of the each individual participant, which would be a future area to potentially explore. But with these projecting onto common brain regions, you can actually pre-compute this and just put it into the network at the right time with the matrix multiplication. And basically what you get is everything before this ROI projection layer is in electrode space, but it doesn't matter because it's just all on the time, you're just operating on the time series, time dimension of the signal. But then once you get to here, everything is mapped onto a common coordinate system. So ROI1 is the exact same no matter what participant you have. And then you can do spatial filtering, all the rest of it, same as what EEGNet already does. And so we called our network, which is basically a modified EEGNet, HTNet, or Hilbert transform net, which is what we use to convert to this frequency domain. And so now that we've talked about how we built the generalized decoder, talk about actually like how it works when we apply it to real data. So testing this on real data, we used ECOG data from 12 participants, where the movements were spontaneous and unstructured over several days. And I want to shout out to Saprit Singh, who is a grad student in Bing's lab, who did a lot of the work for finding these events for these participants. Additionally, we looked at a comparative EEG data set of 15 participants that were just doing a controlled arm movement task. And in both cases, we looked at a very simple decoding task where we were just decoding between move and rest. And so the design of this task is we took 11 of those 12 ECOG participants, put them in the training validation set, tested on the withheld ECOG participants, so going from ECOG to ECOG, and so we call that same modality. And we also tested on withheld EEG participants, so going from ECOG to EEG, which we called unseen modality. Additionally, we actually did a bunch of folds where we randomized which participants were in the training validation test sets, so that way we didn't bias our results to one particular participant. So for same modality decoding, we're going from ECOG to ECOG. These are the results that we found. So test accuracy is on the y-axis with 0.5 indicating random chance, since we're just decoding between two types of classes. And so HTNet are the results from our decoder, and then EEGNet was the original decoder without our modifications. We also compared it to random forest and minimum distance decoders as well. And in all cases, our decoder seems to outperform the others. It's also worth noting too, that because this is training on multiple participants, we actually had to use the projection step onto common brain regions for all of these. So a lot of the performance gains are due to this explicit conversion into the frequency domain. And additionally, our model tends to perform a lot better than others for most participants. Additionally, for unseen modality decoding for ECOG to EEG, the difference is much more striking. HTNet's really the only one that performs above chance and does so for pretty much every single participant. And the reason for this is because it's actually because it's operating in the frequency domain. So this is the example of the difference in ECOG activity averaged across all 12 participants, where we're taking the movement, the average spectrogram during movement, and subtracting it from the rest. And so you see this low frequency power increase and this higher frequency power decrease, similar to what I showed you before. And when you do the same thing for EEG, you don't see this high frequency power increase, but you do see this low frequency power decrease. And what's really notable too is actually the magnitude is very similar. So to me, this really speaks to why it's important to look in the frequency domain, not just keep your signals in the time domain when you're analyzing them. Additionally, so now that I've shown you how our model does pretty well with real data, when we fine tune that model, so this is actually work that collaborated a lot with Zoe Stein-Hansen, another graduate student in Bing's lab. And so when we and so most decoders usually do something like this, where you're operating on the same participant, you're training on them, validating and testing on some withheld data. And you're starting with a randomly initialized model. So the idea with fine tuning is when we actually train a model across multiple participants, we actually get a model that's probably a better starting point than a randomly initialized model. Otherwise, we have a pretty bad model. And so we can use the information from that pre-trained model to fine tune to this new participant. So that's really the idea there. And so when we fine tune performance, test accuracy is on the y-axis again, and on the x-axis is the number of training events used. And so you can see at time 0 or at 0 events, that's the exact same as what I showed you before. So this is just the pre-trained model. But you can see that fine-tuning performance really increases the average decoder performance for HTNet very quickly with just a few number of events. Probably it looks like about 100. And so it does saturate very quickly, but this performance down here is pretty good considering you're only using about 100 events. And without pre-training, it takes much longer, upwards of 400 events for it to really achieve that performance. And you see something very similar with the unseen modality decoding as well. So this is really promising that not only does our decoder generalize better, but also seems to fine tune with just a few number of events in a way that could be really useful for more real world decoding. So just to wrap up this part, talked about building a generalized decoder using explicit conversion to the frequency domain and projection onto common brain regions. Talked about how it sort of outperformed these other state-of-the-art decoders, and that fine-tuning with just a few number of events can really yield nice performance that takes many hundreds of more trials potentially for just a model that's not pre-trained. And one future direction that I'm really excited about with this is taking that model and doing unsupervised clustering, because we have all of this large scale data that's really unlabeled. We have a lot of audio, ECOG, and video recordings throughout several days for every participant. And it was, it's hard enough to get just movement events, but figuring out a way to get some good clustering of this large scale data set would be useful. And one way I'm looking at right now is to do an approach like this, where you take convolutional neural networks and basically use labels that you generate from each to train the other, with the idea that if you have two types of data streams that have shared information, you potentially get a nice, good representation that accounts for both. And so in this case, they're looking at video and audio and looking at audio visual features. In my case, I was interested in looking at the ECOG data we have and also the pose data that SOT used to find those events, those movement events from before. And so the preliminary results I have, at least for move and rest, are pretty promising, but I'm hoping to expand these out into more complex movements than this. The pose model, when actually trained on withheld data, tested on withheld data, excuse me, performs really well. So it's really easy for it to pick up move versus rest with pose. And then the ECOG model too performs, on the order of that supervised model, which might not be surprising given that the pose does really well anyway. But I'm also working with other more complex movements, and the results seem pretty promising. So in summary, I've shown two examples of how you can explore naturalistic neuroscience and naturalistic human movements, looking at neural response to perturbed balance and generalized decoding of unstructured arm movements, where we found this consistent or fairly similar neural response in high-density electroencephalography, excuse me, that was source localized, and for the generalized decoding that significantly outperformed other state-of-the-art decoders and works with electrocorticography where there are non-standard electrode types, positions, excuse me. Okay, and so, yeah, as I mentioned before, there's really a lot of power in leveraging the advantages of neural population recordings in terms of mobility and naturalistic experiments with data science tools that it can account for the limitations of them to really study these naturalistic neuroscience and naturalistic movements. And so we might not be able to study all these kinds of behaviors yet, but we do seem to be trending towards naturalistic neuroscience and understanding what's going on in the brain during more naturalistic movements. And so I just want to thank everyone in the Bing's lab, especially Zoe and Nathan, who worked really hard on that second project I presented, as well as my PhD advisor, Dan Ferris, and his lab, the Human Neuromechanics Lab, which is now at Florida. I also want to thank Raj's lab for help with working with ECOG data, as well as the Harborview staff who helped us obtain and work with the clinical ECOG data, as well as all my funding sources over the years. And thank you all for listening, and I'll take any questions you have.
Great, thank you so much, Steve. It was a great talk. So if you have questions, please drop them in the Q&A. And let's see, as we wait for those to come in, Steve, I was struck by, and maybe I didn't understand this, but it sounded like you said that generally these models are created off a single individual where different sort of, am I understanding that, where you're sort of using the same individual to get the training data as well as the, anyway, rather than getting that, collecting data from, like you were doing with the pre-train, where you're getting it from a number of different individuals. And that's, if I'm understanding that right, that struck me as really surprising given the variability that you showed in just from individual to individual. I just was wondering if you could sort of reflect on that, and if I'm understanding that correctly to be what's been done in the past, and yeah.
Yeah, I mean, so yes, you're definitely right. There's a lot of variability from one subject to the next. And I think a lot of the limitations, or a lot of the reasons for not doing that previously, have to do with not being able to necessarily line up the data that you have. So if you do EEG, it's very easy to do that. But additionally, there's also a lot of variability you get just from trial to trial with ECOG and EEG anyway. So being able to train with something across participants adds another layer of variability, but it also potentially makes your decoder a lot more robust.
So because it seems like if you wanted to, I mean, it sounds like being able to deploy this almost real time to assess maybe what somebody's experiencing. I don't know if that's kind of the end goal, but that you're able to get at that much more readily with this pre-trained data.
Yeah, that's the hope. And I think we were originally planning to do that before COVID happened. So.
Yeah. Nicoletta, did you have a question?
I was about to ask the same question actually, because also like I was surprised by that variability, and just made me think that if there is a difference between men, women, like by gender or age or other types of factors that could explain this variability. I was also thinking like the end goal of the research would be in the end to inform to understand what's happening in the brain, but to inform some treatment options, some like what is the overarching goal?
Yeah, I mean, yeah, I think each of them both of those projects have slightly different goals, where the first one is a little bit more understand the brain and potentially use that as like a biomarker or potentially as a way to develop balance training methods. Whereas the second one is more of instead of viewing and understanding what's going on in the brain, to really just use some of the knowledge we have to just decode it and generate a useful signal and just predict what's going on behaviorally based on the data. Does that sort of answer your question?
Yes, yeah.
And it seems like the second method could be applied across, that technique anyway, could be applied for a lot of different types of areas or different sort of actions or movements. Yeah. I was struck by too, wondering if the cross-modal clustering that you were talking about, if that would, just going back to the balance and how you were contrasting the different types of sensory input that go into balance, if that cross-modal clustering would be applicable for that kind of study.
Potentially. So you're talking about, by cross-modal, you're talking about the second project, like looking from ECOG to EEG?
Well, I think you were like cross-modal, you were talking about like somebody taking audio data and other type of data. I was thinking like with the balance that those are sort of, maybe that's not quite what you were talking about, but sort of two very different types of signals that your brain is getting that are contributing to balance, and trying to understand what the interplay is between them. Maybe I'm not on.
No, that's a really interesting idea. I think the only issue, or one of the main issues with that, would be that you really want to get really nice precise timing when you're looking at balance perturbations and whatnot. And this unsupervised clustering is kind of, I don't know, a little bit more of like brute force and a little probably less temporally precise than you would want from like, when do they start to lose their balance, or when did I give them this perturbation or something. Okay. At least as far as I know with the current technologies.
Yeah, that makes sense. Did you have a lot of fun watching people trying to balance on the balance? I kept thinking how many people fell off, but I was glad to hear that you had a way that you constrained them and kept them upright.
Yeah, I got to try all of these too, so
yeah,
that was my own guinea pig.
Let's see, we have a question that just came in. Thank you for an amazing talk. Are the brain regions ROIs used in the work so far defined based on some brain atlas template, or are the ROIs defined in a data-driven way, for example, with unsupervised clustering?
Yeah, that's a great question. So for this, we just used a pretty basic atlas. So we used positions. So originally we used the AAL atlas, but we actually used this parcellation of the MNI brain, into about 3,000 really tiny brain regions to get a little bit better spatial resolution. But one of the things I was interested in kind of extending this work potentially is to sort of use that as a starting point. So have this sort of common regions that you base your starting point off of, and then actually learn a better representation, because it's a very simplified idea. It has no idea what's going on in terms of like sulci and gyri. It really doesn't account for any specific anatomical differences from one participant to the next.
Well, thank you so much, Stephen. I think we'll wrap up there, and I'm sure you're happy to take questions if people have things that come up and they want to reach out to you. And thank you again for the seminar today and for leaving us with this beautiful picture, which looks a lot more like summer than our current winter in Seattle. So thank you for that light and beautiful shot, and for a lovely talk. So thank you all, and we'll see you all next week.
Thank you.
Thanks for having me.